All right. Well, thanks very much for the invitation to present on what I think is sort of a new and emerging and really exciting topic in transplantation. And that's sort of using technologies that are constantly improving to understand how we can really find donors and recipient pairs that are optimally suited for one another so we can maximize the outcomes of transplantation. So I'm going to talk in 15 minutes about a relatively complex topic known as molecular matching or eplet matching. So to answer the question as to what are eplets, we kind of take a dive back into a basic biology textbook where we see a schematic of a cell. In the cell's nucleus, we have the chromosomes of the individual, and we'll focus on chromosome 6 here, which houses 6 very important genes known as the human leukocyte antigen genes, or HLA genes. They're A, B, C, D, R, D, P, and DQ. And these, the genotype, so what you express at these 6 different gene loci are sort of what make up your genetic fingerprint, what makes every individual unique. And it's relevant to talk about all this because it sort of plays into why matching between donors and recipients is so complex. So these genes are what are referred to as highly polymorphic, meaning that there are multiple different varieties of expressions of these genes. And if you equate it to eye color, you can think of there's really only a couple different expressions of eye color. There's blue, there's brown, there's green. But if you look at the HLA genes, so the ABC, DRDP, and DQ, what are listed here are the really very creatively named alleles. So that in the A locus, you can have an A1, A2, A3, et cetera, et cetera, et cetera, and you get the idea. But there are a great, extraordinary number of potential alleles that individuals can express at each one of these genes. And so you start to understand how the combinations of these can be really nearly infinite. So we do know the genotypes of all donors and all recipients in transplant. It's part of what we do in the transplant workup. So if you look at this, and this is some actual typing from a donor, we get a report that looks like this. So if this is a recipient, we know that this is, so their genotype. So we each have 2 copies of each one of these genes. So here, this person is an A30 and an A74, a B42, a B53, and so on and so forth. And so we can compare recipient genotypes to a potential donor genotype. And so if you look at this, here we have the recipient with this genotype, and lo and behold, this donor is an A30, an A74, a B42, a B53, and working your way down the line, you can see that every single one of these is matched. So this is sort of the coveted perfect match. And so this is what we all really want. So how often does this really happen in transplantation? Well, this is how often it happens. It happens when the donor and the recipient are identical twins. So extraordinarily unrare scenario, the perfect match between HLA, between donor and recipient. So assuming this recipient doesn't have an identical twin willing to donate to them, they come forward with a different donor. And here we have an A1, an A68, a B7, a B13, and so on and so forth. Not a single one of these HLA alleles is matched. So is this donor unacceptable for the recipient? No, it's not unacceptable at all. So not matched doesn't mean not compatible. And so why does it even matter? Why do we even care about HLA matching if I just told you that you can be a perfectly acceptable donor if you're not matched at a single HLA locus? Well, in the simplest terms, we can think of every mismatch as a potential opportunity for rejection. So one's immune system is going to very stealthily and very accurately identify the HLA that is not their selves, and that will serve as a target for an immune attack. So each one of these mismatches is a potential opportunity for the immune system of the recipient to respond and potentially reject the kidney. And so this is why we care about HLA match. We mitigate those mismatches with immunosuppression, but it sort of stands to reason that the fewer opportunities there are for the immune system to reject a mismatched organ, the better that scenario is, and maybe the less immunosuppression that we need. So if you take this recipient here and you compare 2 potential donors for this patient, here I've circled the HLA loci that are matched. So here donor number 1 has 5 out of 6. And when I say 6, I'm talking about A, B, and the DR genes. So 5 out of 6 of these antigens are matched compared to donor 2, where only 1 out of the 6 are matched. So intuitively, you might say this is probably the better donor, the 5 out of 6 match versus the 1 out of the 6 match. And I thought Massimo should be the one giving the talk, so I gave him a few words here. Massimo would tell you, he's sitting over there, so if there are questions about this talk, please direct them to him. He's our HLA guru here at NYU. And what Massimo would tell you is this is not as simple as I've even outlined it so far. He would tell you that in certain scenarios, in fact, in many scenarios, an A2 that seems like it's matched isn't even an A2. This is sort of a low resolution nomenclature. And we know that among the A2 antigen group, there are multiple different A2 genes or A2 alleles. So you can have A201, you can have A202, you can have et cetera. But that's not it. You actually have all of these at just the A2 antigen group. So there's tremendous variability in differences between what genes are expressed by certain individuals. And so this is where the EPLET terminology sort of comes into the conversation. And so what are EPLETs? Well, it's sort of a different way about thinking about how we describe the different genes that people express at all these loci. So EPLETs are little structural subunits of the actual HLA molecule, which is here sitting on the cell surface. And when we think about how we characterize HLA molecules in terms of EPLETs, we think about the different constellation of clusters of these little structures that each one of these molecules expresses. So we're kind of comparing shapes of molecules rather than these sort of generic names. So looking at this on a more sort of molecular level, if this little blob right here is an HLA molecule sitting on the cell surface, what I've highlighted here in teal is basically one little EPLET. So EPLETs are defined as short stretches of polymorphic residues or amino acids that are located in accessible regions. So on the cell surface, where a recipient immune system molecule could sort of recognize and interact with this cluster. They happen to be contiguous in three-dimensional space, so they're not necessarily right next to one another in linear sequence, but when the protein folds, they kind of come together and form this cluster. And most importantly, and why any of this is really relevant, is this is all totally predictable by HLA gene sequencing. And we now have the ability to very rapidly perform sequencing across HLA genomes. And this is what is starting to be done very routinely in transplant. And so when we do EPLET matching analysis, it really is as simple as sequencing across the donor and the recipient HLA, using the genetic sequences to predict the groupings of EPLETs that genotype expresses, and then comparing them. We can identify the EPLETs that are present in the donor that are not present in the recipient as targets for the recipient immune attack. So we basically count and compare. And so this sort of illustrates on a very distilled down level how we do that. So here we have a recipient that has this genotype DQ8 and two potential donors. So we know that they're not matched at this locus, but if you look at this single antigen mismatch, a DQ8 versus a DQ5, and you count the EPLETs that are mismatched, well, here there are 27 EPLETs that are mismatched. So 27 spots on this protein that structurally look different from the recipient's protein, compared to this other potential donor where there are only two. So if you talk about it in the context of opportunity for immunoreactivity or rejection, this is the far better donor. There are only two opportunities for the immune system, the recipient immune system, to sort of target or attack this molecule. And so this is what EPLET matches is. We can do this at every single one of the genes, and we can kind of come up with a score that constitutes the mismatch between a recipient and a potential donor. And we now know very clearly, very convincingly, that EPLET mismatch correlates with risks for rejection. So just want to highlight one paper that's probably the most cited by the person who's done the most work in this area, which is Peter Nickerson from Winnipeg, Canada. So this is really a landmark study where he looked at patients and sort of grouped them based on their EPLET mismatch score into three groups. So there's a low risk, intermediate risk, and a high risk group, defined by these thresholds that we won't talk about how he figured them out, but suffice it to say, he sort of drew these lines to put patients into these three categories. And they defined the low risk category as having less than 7 mismatches, EPLET mismatches at DR, and less than 9 at DQ. And then when you look at how patients do with respect to rejection, there are very clear stratifications for how these different groups perform. So the green line here are the low EPLET mismatch, the quote unquote low risk group. And what we're looking at here is T cell rejection free survival. So these patients live longer with less T cell rejection compared to the intermediate or the high risk group. And if you look at antibody mediated rejection, the picture is really the same, with this sort of stepwise decrease in outcome or stepwise increase in antibody-mediated rejection-free survival as you decrease the number of mismatches. So the lowest mismatch patients do the best. And most importantly, I'm going to gloss over the data here. What this sort of all translates to, and it intuitively makes sense, is that these patients also probably need less immunosuppression. And we've heard some really powerful stories from patients today about what immunosuppression means in terms of living your life. This is the biggest double-edged sword in transplant, right? It prevents rejection. It allows us to do transplants at all, and it allows short-term survival to be pretty good. But there are a lot of other downsides that are very significant that impact patients' quality of life and ultimately how long patients live and how long their grafts are going to function. And so now that we have the ability to, at a very sophisticated level, compare donors and recipients, that we think we can sort of gauge what are the highest risk and the lowest risk pairs, let's choose now to do better matched transplants that need less immunosuppression and can function better for longer. So I just want to sort of briefly talk about how this sort of new technology is being put into practice and being used today. And I think we'll see this expand over the coming years. So this is just an image I grabbed from the National Kidney Registry webpage. I'm not a spokesperson for the NKR, but we do participate in the NKR. And I did get permission from the NKR to show some of these images. The NKR, under the leadership of Garrett Hill, its founder, who himself is a kidney donor, has not only become the most prolific paired kidney registry in the United States and actually facilitates, it's probably now closer to a quarter of the living donor kidney transplants in the US. They've also been extremely innovative about integrating new technologies. And so, as we heard really nicely from Dr. Goldstein, this sort of methodology was originally conceived to overcome incompatibilities between donors, where donors are swapped in order to make sure that two incompatible pairs become two compatible pairs. Well, the NKR now is using this registry, this clearinghouse that they have, to really examine the potential matches between donors and recipients, not just to find compatible organs, but to find the best matched organs or the lowest eplet mismatched pairings. And so that is the foundation of the Kidney for Life initiative, where donor and recipient pairs, living donor and recipient pairs, who are willing to go into this exchange registry, not just to get a compatible donor, but to get the best matched donor, are doing so. And potential pairs, everyone is genotyped, and there's an ongoing in real time process whereby pairs are analyzed for various criteria. You look at age, you look at size, you look at HLA antibodies, but you can also look at eplet matches, and this can all be balanced with the ability to maximize the number of pairs that are generated out of this pool of organ donors and recipients. And this has been the outcome of that, which is really quite striking. So 2020 was when this idea was first conceived of and proposed. And these are the numbers of low eplet mismatched living donor transplants that have been done in that time. And so in 2024, an estimated 1/3 of all of the living donor transplants that have been facilitated by the NKR are projected to be low eplet mismatched transplants. And so the Kidney for Life, kidneyforlife.org, this is the webpage where you can go to get information about how to sort of register for this. There are so many transplant programs across the country that participate. So the message to patients is, think about this, ask your teams about it. I think there's going to be a lot of really, really powerful data that you start to see come out very soon. We at our site are starting to do some clinical trials of how we can minimize immunosuppression safely in these patients. And the data are a little bit too preliminary to show right now, but I think what we've seen so far is extremely encouraging. So that is your crash course into molecular matching. And thank you for the invitation to speak, and thanks for your attention.